Welcome to our symposium this morning, Putting the Pancreas on the Pedestal. I am Rebecca Hull-Mikely from the VA Puget Sound and University of Washington in Seattle, and I'm very pleased to co-chair this session with Zach Sellers. Before we get started, I wanted to just give you a little bit of housekeeping announcements. We're going to do, I don't know if all of the symposia will be like this, but this morning we're going to do each talk and then questions after the talks, okay? I think they're perhaps distinct enough that it will work better that way than doing a panel discussion. Also, if you happen to have the apps open, you can enter Q&A into the app. That's the way that the system has kind of asked us to do it. You will notice that there actually aren't floor mics, but if you feel the burning desire to get up and ask a real question to a real human being, please go ahead and do that, and we will be monitoring the iPad. Don't get me wrong, we're technologically capable, but if you want to get up and ask a question, please do, and we'll go ahead and repeat it. Zach has a couple introductory slides, so I'm going to pass it over to him.
Thank you, Rebecca, and welcome everyone. It's great to see everyone, and it's really a pleasure to co-chair this session. And I like the title that they gave us, Putting the Pancreas on a Pedestal, because as a pancreatologist, I really think it should be. So just have a few introduction slides, and then we'll dive into the talks. I think we have some really great talks for you today. So here are the learning objectives for our session. We're going to talk about some of the transcriptomics research that has been emerging and what it's telling us about the endocrine and exocrine components of the pancreas. We're also going to talk a lot about the anatomical and functional interplay between the exocrine and endocrine pancreas, both in health and CF. And I think historically these are two areas that have been handled by different types of providers, and there hasn't historically been a lot of discussion between them. But I think that's changing, and I'm excited for that to change. And then, of course, we're going to talk about the current research and what we know and what we don't know on the potential impacts of CFTR modulators on both exocrine and endocrine function of the pancreas. So this is a slide I often share with trainees and others when I talk about the pancreas, and it has a couple, like a cartoon and actually histology, but what I like to stress is that really the endocrine and exocrine components are really enmeshed together, right? Again, we often think about them separately. As a gastroenterologist, I tend to think about the exocrine components, but the islets here, as you can see, are really surrounded by these acinar lobules. And so it's naive of us to think that they are not going to influence each other. And in fact, I think there's some interesting data that's come out and shown that really the blood supply between the islets and exocrine components are enmeshed and they communicate with each other. And I think that will be a theme of what you'll see today. And then in regards to function, I really like the study done by Dr. Koushik Moon, currently at Cedars-Sinai, where he used a microfluidic device and separated out the pancreatic ductal cells and the islets. And he could show that if he perturbed the ductal cells, he could act with and inhibit CFTR, it would actually affect insulin secretion from the islets without those islets ever seeing the CFTR inhibitor. Again, so I encourage you to look at this paper and other papers, but I think it points to this also not only anatomical but functional interplay between the exocrine and endocrine components of the pancreas. So we have 4 wonderful talks today. We're going to talk about transcriptomics, and that's going to be by Kyle Galton. We're going to have the interplay between the exocrine and endocrine pancreas by James Shaw. And then we're going to talk about CFTR modulators on endocrine pancreatic function by Andrea Kelly, and CFTR modulators on exocrine pancreatic function by Vikesh Singh. And as Rebecca said, please ask your questions. We want this to be very interactive. We got a great group of speakers today.
Okay, so our first speaker is Kyle Galton. Kyle's an associate professor in the Department of Pediatrics at UCSD, and he is a leading expert in computational biology and analysis of the pancreas. It's really a pleasure to have him here today. And Kyle's going to talk to us again this morning about what are we learning from the pancreas in single-cell genomics.
Okay. So thank you so much for the invitation to speak and also for the opportunity to be in my hometown for the World Series, although it didn't go well last night. We'll see. I might be bad luck for being here, but anyway, that's been quite fun. So yeah, like Rebecca said, our lab studies the pancreas and the regulation of cell types in the pancreas primarily through the lens of diabetes, type 1 and type 2 diabetes, although after working with Rebecca, we've started thinking more about changes in the pancreas in CF-related diabetes. Okay, so how do I change the slides here? There you go. Just click. Okay. And here's my disclosure. Okay, so generally, the identity and function of cell types in human tissues is determined by the regulation of gene activity. This is primarily controlled by what I'm calling cis-regulatory elements, which are sequences in the human genome, such as promoters and enhancers that drive gene regulation and gene expression. And that's primarily how you get differences in function and responses of cell types to certain stimuli is by activation of cis-regulatory elements in different parts of the genome. In many cases, these regulatory elements can be specific to certain cell types and drive changes in gene expression specific to those cell types. And by and large, this is how cell types acquire their unique identities and functions. The way that we measure regulatory activity is in our lab, at least, is using high-throughput genomic and epigenomic assays. And so I've listed some of the assays that are used to profile not only cis-regulatory elements in the genome, but also relationships between these elements and genes that they regulate and the production of gene transcripts. I've highlighted two assays in particular that I'll be talking about today, which are the primary assays that we focus on in our lab. One probably familiar to some, if not many of you, which is RNA-seq, which is a measurement of gene transcripts using high-throughput sequencing. And then the other assay that may not be as familiar to some of you is called TAC-seq, which is an assay that measures what I'm calling accessible chromatin, which is a hallmark of regulatory activity, which allows you to define the location of cis-regulatory elements. And so this is just a more in-depth overview of accessible chromatin. It's also called open chromatin. So again, this is an epigenomic hallmark of active regulatory activity. Normally in the genome sequence is in a closed configuration wrapped around nucleosomes and inaccessible to regulatory proteins that may come and bind parts of the genome. Those nucleosomes can be displaced or removed, and that opens up the genomic sequence, then allowing transcription factors and other DNA binding proteins to come interact with the genome sequence and drive regulatory activity. And so assays such as a TAC-seq exploit this phenomenon whereby you have basically open stretches of DNA. The way that a TAC-seq works is that there's a transposase, which is hyperactive, which is loaded up with basically barcodes for sequencing. And this transposase will preferentially integrate these sequencing primers into these naked stretches of DNA, which mark regions of open chromatin. And in the bottom right, you can see by doing this assay and the results of this assay, you can mark where cis-regulatory elements are, both promoters at the start of a gene as well as elements like enhancers, which can be far away from, in some cases, the genes they're regulating. And traditionally, these assays have been done in bulk, and by that I mean you take a heterogeneous tissue and you run the assay across all the cells in the tissue, and you get kind of bulk level profiles describing the gene regulatory programs of the entire tissue. But since, as I mentioned earlier, the regulation of each individual cell type is highly specific to that cell type, what we really want to understand is the regulatory program, accessible chromatin sites, gene expression in each individual cell type. And of course, as was mentioned in the intro, the pancreas is a heterogeneous tissue comprised of numerous different cell types. You have the endocrine and exocrine components of the pancreas, as well as other cell types in the pancreas, and dysregulation of different cell types in the pancreas leads to a variety of different diseases: diabetes, pancreatitis, pancreatic cancer, monogenic diseases such as maturity onset diabetes of the young, or MODY, as well as CF-related diabetes. And so that's our goal, is to understand how dysregulation of individual cell types contributes to these diseases. And recently, the advent of single-cell assays has enabled us to profile the cell type-specific regulation of individual cell types within heterogeneous tissues such as the pancreas. Some advantages of single-cell approaches over bulk assays, as well as, say, like FACS sorting individual cell types, are that first you can separate out the component cell types of a tissue in a single experiment. You can reveal both discrete as well as continuous heterogeneity in, well, say chromatin accessibility, but gene regulation within a cell type that might not be as easily identified by, say, FACS sorting-based approaches. You gain large numbers of independent observations of cells. So in our assays, we tend to generate data from 10 to 20,000 cells, and this enables you to do really interesting correlative analyses across cells. And then it also has nice features in terms of being able to pool many samples in an assay and then deconvolute those samples based on the genetics of each individual cell. So the two assays that we primarily use in our lab allow us to profile ATAC-seq, again, which is accessible chromatin, and RNA-seq, which is measurements of gene expression. There are multiple different flavors of these assays that we've used. The first on the left is called combinatorial barcoding. And the way that this assay works is that you take a tissue, you isolate nuclei from that tissue, you distribute those nuclei into wells, and then you perform the assay, in this case ATAC-seq, in each individual well, where each cell in that well will get one barcode. You then reaggregate the cells, redistribute them, and then add a second barcode via PCR, and therefore every cell gets two barcodes, and for the most part, those two barcodes are unique identifiers of every cell, although there's some cells that get the same barcode. And we use computational tools to try to remove those so-called doublet cells. This gives us unique barcodes for the most part for each individual cell. The second on the right are probably more familiar to some of you who have started doing this work, which are droplet-based assays. These are now kits that are being sold by a company called 10x Genomics, and the assay is done in droplets using microfluidics. On the bottom, we're also increasingly performing assays, and I have some data here, but won't talk on this too much, about what's called multi-ohm assays, which enable you to profile both the gene expression as well as the accessible chromatin profiles from the same cell. So from each individual cell, you get both of those profiles, and that allows you to link the regulatory element activity to gene expression within each cell directly. So by doing these assays, we gain insight into numerous aspects of how cell types in tissues such as the pancreas are regulated. So on the top left, the results of the assays look like this, at least for ATAC-seq, where you have regions of the genome, and then you have on the x-axis each individual cell, and then you have reads that you identify from each individual cell. There's a lot of computational work that goes into translating those individual cell profiles to what you see on the bottom left, which is a clustering through which you can define individual cell types that represent specific clusters from your assay. From the definition of cell types, then on the right, there are numerous things that we can define from each cell type, so gene expression levels of the cell type, cis regulatory elements, transcriptional regulators of gene activity, relationships between regulatory elements and the genes that they're regulating. This is, for those of you who don't study genomics, is actually quite a major challenge to link cis regulatory elements to the genes they're regulating, because they can often be quite far away in the genomic sequence from the genes that they affect, as well as on the bottom, signaling networks between different cell types. Another way that we use this information in our lab is to understand risk of disease. Again, primarily we study diabetes, but again, we're increasingly interested in CF-related diabetes. And all these diseases are complex, so they have many genetic risk factors that affect their risk. And determining the function of these variants, given that they're mostly in non-coding parts of the genome, can give us critical insight into how these risk variants are acting and tell us something about disease processes. So if you have a locus that's associated with a pancreatic disease without any annotation of the genome, you might not know anything about how these variants function, but by defining cis regulatory programs in each cell type, you can use this information to define disease-causing elements, genes, networks, and cell types, and so forth. And this, again, can give us insight into how these variants are functioning. Okay. Alrighty, so I'll talk quite briefly. I'm already running low on time, so I will probably very briefly go through both of these. The first project is defining cell type regulatory programs in the pancreas and then also annotating pancreatic disease risk variants, and this was published in Nature earlier last year. And so in this study, our goal was to understand the cis regulatory programs of cell types in the pancreas as well as in the peripheral blood. So we performed these single-cell ATAC-seq assays in whole pancreas samples as well as peripheral blood. And I'm skipping over all the processing steps and just showing the endpoint of the clustering here, where we have over 130,000 nuclei from these experiments, which we can cluster and then annotate which cell types these clusters represent. It's not always so straightforward to define what a cell type represents from ATAC-seq data. So the way that we annotate what cell type we believe a cluster represents is by looking at the promoter of genes that are known to mark the identity of certain cell types, or in some cases, the gene body of genes known to mark cell type. And genes that are highly specific to certain cell types give us clues as to what that cluster might represent. So for example, for one cluster that had high activity, the promoter and gene body of CFTR, and so we know that since CFTR is a highly specific marker of pancreatic ductal cells, that cluster represents ductal cells. So that's how we annotate each cell type. Having defined each cell type, we can then, oh, you can't see the mouse here. Having defined each cell type, we can then, that's right. Yeah, that's right. Let me see actually if I can, there we go, okay. We can take the profiles of each cell type and then call cis regulatory elements using these ATAC-seq profiles within each cell types, and this gives us a map across the genome of where cis regulatory elements are located in each of these cell types, and in doing this we identified about 450,000 cis regulatory elements, many of which are highly specific to one cell type relative to another. We also created a matched map of gene expression using single-cell RNA-seq in the same tissues. We identified basically the same cell types and enable us to correlate the profiles that we observed from ATAC-seq to those from RNA-seq and link cis regulatory activity to gene expression. So one thing that we did with the data was to take those 450,000 sites that we identified and ask which are very specific to one cell type relative to another. And so of the 450,000 sites, about 25,000 were highly specific to one cell type in their profiles relative to other cell types. If you look at these sites that are highly specific to a cell type and ask which genes they're nearby, these sites tend to be proximal to genes that have highly specific functions in that cell type. So for example, for the sites that are specific to ductal cells, we see that these are enriched for proximity to genes involved in water transport and transport of small molecules and processes related to ductal function. Sites specific to acinar cells are proximal to genes involved in digestion and trypsin and so forth. So genes that are marking processes that are highly specific to that cell type, and these sites are also proximal to genes that tend to be much more highly expressed in that cell type relative to others. So we have sites that are active in each cell type, and then sites within the sites that are specific to one cell type relative to another. We also use this information to define transcriptional regulators of each cell type. So for example, in ductal cells were highly enriched for sequence motifs for the HNF1 family. And then if we look in that family for genes in that family, so there's 3 family members in HNF1, HNF1B is highly specifically expressed in ductal cells and therefore confirms that HNF1B is a specific regulator of ductal cells, which is already known. But in many cases, we find novel regulators of activity of different cell types as well as different subtypes of cell types. As I said earlier, a big challenge in understanding the function of cis regulatory elements is annotating the genes that they affect, so we can use single-cell profiles to predict the genes that are affected by cis regulatory element activity within each cell type. And so we did this. In many cases, the genes that are predicted to be regulated by these sites are quite distal, in some cases over a megabase, a million bases away from the gene that they're predicted to regulate. And also many of these links between cis regulatory elements and their target genes are specific to that cell type. And again, the genes linked to these sites tend to have more cell type specific expression for that cell type relative to other cell types. So across all this information, we can define cis-regulatory elements in each cell type, regulators of these elements, and also the genes being targeted and their expression levels of cis-regulatory elements, and this gives us a catalog of cis-regulation in each pancreatic cell type. One thing I mentioned in the introduction is that one of the key areas that we use this information for is to annotate risk variants for complex diseases of the pancreas. And in this study, our primary focus was type 1 diabetes, although it just so happens that one of the key findings from doing this is something related to this audience, in that we found generally that many risk variants for type 1 diabetes fell in sites that were very specific to acinar and ductal cells, which itself was a noteworthy finding, because the role of the exocrine pancreas in type 1 diabetes had not been fully appreciated. But if we looked at which loci that these variants fell at, one in particular was quite interesting to us, in that risk variants for type 1 diabetes, which you can see here, mapped in a region that was intergenic between what's our upstream of the CFTR gene, and mapped in a cis-regulatory element that was highly specific to ductal cells relative to other cell types. This variant also disrupts a motif for HNF1B, which I mentioned before is a ductal-specific regulator, and is also predicted to regulate the activity of CFTR. We confirm this using a technique called CRISPR-I. Basically, you inhibit the activity of this site and then show which genes it affects, and it has very specific effects on CFTR expression. And these variants were also associated with CFTR expression in the pancreas. So it suggests that regulation of CFTR in ductal cells is somehow contributing to risk of developing type 1 diabetes, and also potentially suggests a broader link between type 1 diabetes and pancreatic disease related to cystic fibrosis. So how does CFTR and exocrine cells contribute to risk of type 1 diabetes? So many of these same variants are also associated with pancreatitis and pancreatic cancer. There have been several studies that have suggested that CFTR likely affects CF-related diabetes via inflammation and immune infiltration. In particular, there was a paper in JCI from Al Powers that argued that it was not an intrinsic defect in beta cells, but likely something external, and showed that it was a possible mechanism through immune infiltration. And so it's hypothesized, at least, that a similar mechanism could be contributing to risk of type 1 diabetes, although there's, of course, other possibilities as well. And so again, this example here, that we have numerous examples, but obviously this is a very germane example for this audience of how we can use these maps to understand how risk variants for disease may be affecting cell type specific functions, and in this case uncovers a potential role for CFTR in ductal cells in type 1 diabetes risk. Okay, so that's the first part of the talk. So the second part of the talk, which I will go through very quickly, is now understanding changes in the regulatory programs of pancreatic cell types in pancreatic disease. Again, the focus here is mostly on type 1 diabetes, but hopefully you can see how this paradigm could be applied to CF-related diabetes. And so some of the limitations of the previous study were that they were all from non-diseased individuals, so they were kind of reference maps of each cell type, and they were also from a relatively small number of donors. So in this study now we've expanded the samples that we're profiling to 33 samples, and these samples come from a biorepository called NPOD, which is run by Mark Atkinson at the University of Florida, mostly a type 1 diabetes focused resource, so they do actually have some CF diabetes samples, and Rebecca and I have been working on trying to profile these samples. And so here's again showing you the map of each cell type that we identified from these 33 samples clustered together. One of the noteworthy findings here was that not only did we identify cell types in the pancreas, but we also now, with the resolution of samples and number of cells, start to identify potential subtypes of each cell type. In particular, we identify numerous potential subtypes of exocrine cells, acinar, and ductal cells. That's the RNA map, and then now here's the ATAC-seq map, which are from the same samples. So we have gene expression levels from RNA-seq from each cell type and subtype, and then again, accessible chromatin profiles from the same cell types. But again, we were quite interested in this potential heterogeneity that we observed in exocrine cells and its potential role in disease. So for acinar cells, we identified 3 potential subtypes of acinar cells. One has been characterized previously called Reg-positive acinar cells. And these cells are enriched for genes that have the pancreatic enzymes that are produced by acinar cells, so they have much higher expression in these Reg-positive cells relative to other acinar cells. We also see other subpopulations of acinar cells, one of which appears to be a signaling response, potentially inflammatory-related subpopulation of acinar cells. And this suggests that while it's the same cell type, that these are different subpopulations of the same cell type that we're identifying from these assays that you may not have identified via bulk assays or by FACS sorting, which may give you insight into the functions of these cells. And this is just showing you for the Reg-positive acinar cells that they have increased levels of many enzymes and other highly produced genes in acinar cells. These subtypes of acinar cells are also enriched for different transcriptional regulators. So for the signaling subtype of acinar cells, again, related potentially to inflammation and cytokine signaling, they're also enriched for motifs related to inflammatory response in the cells STAT and IRF. So that gives us just the resolution in terms of samples and number of cells, gives us insight into potential subtypes of these cell types, but then we're again interested in understanding how these things change in disease, and our focus here is type 1 diabetes. And so of the 33 samples we have, some are controls, some are individuals who are positive for autoantibodies, so they did not develop type 1 diabetes, but they had multiple autoantibodies, and therefore were almost certainly would have developed type 1 diabetes at some point in their life, and then also individuals who had type 1 diabetes for varying amounts of time. And so very quickly, some insights here. So we see broad changes in the proportions of cell types in the pancreas in not only type 1 diabetes, but also in the autoantibody positive individuals, reduction of beta cells, of course, because these are the cells that are destroyed in type 1 diabetes, but also an increase in immune cell populations, macrophages, CD8 positive T cells, and some potential evidence for changes in even subtypes of these cell types. So we have an increase in the Reg-positive acinar cells in the early stages of type 1 diabetes, although this was not a significant change. Within each cell type, we also find, in some cases, quite dramatic changes in the gene regulatory programs and genes that are expressed in each cell type, to a lesser extent in the autoantibody positive individuals, but in even the early stages of type 1 diabetes, not just for beta cells, which you would expect because those are the target of immune attack, but other cell types in the pancreas as well, suggesting that there are more coordinated, broader changes occurring in the pancreas in type 1 diabetes. Supporting that, we also find changes in predicted signaling between these cell types. To define cell signaling networks, we use methods that define things called ligand receptor pairs, where basically you measure the expression of a ligand from one cell type and the expression of the receptor in another cell type, and you predict relationships between cell types based on these ligand receptor pairs. And then again, we ask whether these relationships change in disease. And so generally, we see a change in the kind of total signaling between cell types at the high level across all cell types as you go from non-diabetic to autoantibody positive and early T1D. We also see changes in both the outgoing signals and the incoming signals for specific cell types. Beta cells, again, of course, we see an increase in outgoing signals from immune cells in early stages of T1D, endothelial cells, which have been suggested to play a role in type 1 diabetes, but also changes in exocrine cells as well. Okay, so I'm going to skip the last parts just in the interest of time, and just, because I'm thinking about time, and just say a couple last points. So one of the things that our lab spends a lot of time doing is not only generating these data and interpreting them, but also trying to provide them to the community. So we generate databases, basically, that host a lot of these data, working closely with the Broad Institute, as well as a consortium called the AMP, Accelerating Medicines Partnership Consortium, which includes members of pharma, where we provide these data to the community from genomics, which can be, in some cases, quite complex to work with and interpret, not only to visualize these data, but then also to use them in concert with, say, human genetic data to identify new drug targets and understand where genes may be functioning, which cell types they're active in, and which genes may be good candidates for furthering drug discovery. And so these resources are freely available for anybody to use, not just within the consortium, and allows you to access these data and interact with them. Okay, so in summary, using single-cell genomics allowed us to reveal cis-regulatory programs in a wide diversity of cell types in the pancreas. Not only exocrine and endocrine cells, but other cell types as well. Pancreatic exocrine cell types, as well as endocrine. I didn't have time to talk about this, but we have other studies that have shown this as well for beta cells and other endocrine cells, that there are discrete subclusters which likely represent distinct states of these cells, which have unique genomic properties and in some cases have differences in their profiles across disease. Loci that affect pancreatic disease impact cell type specific regulations. For example, variants of CFTR affect ductal specific regulation and may contribute to type 1 diabetes risk. And then using these assays, we can drive changes across disease, such as type 1 diabetes. But again, like I said, we're interested in doing similar studies in CF-related diabetes to give you insight into the cell types that are driving disease. So thank you very much, and I'm happy to take any questions.
Thank you, Kyle. That was great, as always. Those of you at the back, there are a couple of seats up here if you want to come and sit rather than just standing up for a couple hours. First question, thank you for using the technology, is from Katie Larson-Odie. She asks, can you speculate as to the importance of macrophages and any relationship to CFTR dysfunction? Not show up at all.
In CF-related diabetes?
Just, I think in general. Any link between CFTR and macrophage function in the pancreas?
I do not know. I mean, we find pretty specific expression of CFTR to ductal cells. What happens external to the cells in regards to signaling to other cell types. And we do see a lot of signals emerging from ductal cells, again, in type 1 diabetes, perhaps in CF-related diabetes as well, that signal to a variety of different cell types. I don't, off the top of my head, know exactly what the relationship is between ductal signals and macrophage function. And we do see quite marked changes in macrophage populations in the early stages of type 1 diabetes, like I mentioned. So that seems relevant, at least to type 1 diabetes. But the relationship to CFTR in that regard, we have not looked specifically.
Yeah, go for it.
Great talk, Kyle. Just a practical question. When you do these studies, how much tissue do you need, and how can people start to think about, contributing to this?
You don't need that much tissue. So we typically require like a million cells to do these assays. I don't know what that would equate to in terms of a tissue amount. But the tissue that we get, say, from NPOD, we get quite a small amount of tissue from them. And when we take like purified islets, we do a lot of studies on purified islets. We usually take like, say, several million cells, like thousands of islet equivalents, and are able to run the assays from relatively small numbers of cells. Like bulk assays, you could actually, like for ATAC-seq in particular, you can actually do as in little as, say, like 10, 20,000 cells. So even less. Single cell assays need a bit more than that, but still a million cells is tractable for many applications. And Rebecca and I were just talking about this earlier. We're also starting to try to use these assays to profile FFP, formalin-fixed tissues, which may unlock like many samples that have been inaccessible to these assays. There are new kits that allow you to run these assays from FFP samples, which again, so far seem like they require similar small amounts of starting material. So.
One quick question for me before we move on. So with the ATAC-seq that you've got, one question that's been kind of sort of going around in my brain is that CFTR itself is a big complex gene, and it seems to be regulated in a very different way in different tissues. And our understanding of how it's regulated in the pancreas is actually quite poor. And so I don't know what the resolution is of the data, especially from the single cell assays that you have, but do you think that there's enough resolution to shed light on where cis-regulatory elements might be in the pancreas for CFTR?
So we know where the regulatory elements are located. which, if there's multiple isoforms, say, of CFTR, which isoform they regulate, that's actually a bit of a challenge. So one limitation of at least the 10x assays for gene expression is that they tend to be profiling just the one small part of the transcripts. You don't get like full-length transcripts. There's other single-cell assays like SmartSeq and different technologies that you can actually do like full-length transcript profiling from individual cells. These assays just give you basically tags for the genes. So you can't actually look that in depth at expression of different isoforms. So that is one limitation. But from the ATAC-seq data, you can determine the location of regulatory elements throughout the gene, and we've looked at this and where regulatory elements are throughout the locus. You can maybe predict which promoter they're affecting. So if there's different promoters of CFTR or other genes, you can get some insight into are they regulating an alternate promoter, things like that. I think beyond that, like where the regulatory elements are, and probably more depth interrogation by modulating those elements and in cell models and stuff, and seeing what their effects are in CFTR regulation would probably be a next set of steps that you'd want to take to understand really like what they're regulating at the locus. Yeah.
Okay, great. Awesome. Okay.
Okay, thank you.
Very good. Thanks so much, Kyle.
Okay. Moving on, our next speaker is Jim Shaw from the University of Newcastle in the UK. So he's at the right end of the day for his jet lag. Let's see. I think, is that the first slide of your talk?
Yes.
Oh, fantastic.
I tried
Okay.
to be well behaved.
So Jim has been working in this area for a while. He's known very much for his work in type 1 and islet transplantation, but has some really new and cool data to share in terms of looking at CF pancreas. Jim, go ahead.
Thanks very much. Good morning, everyone. Thank you to both inviters for asking me to come and speak in this wonderful meeting in this great city. I'm just beginning to learn a tiny bit about the World Series, but press. Okay, thank you. Oh, fantastic. I've got no relationships to disclose relevant to this presentation. So we're taking a very unashamedly clinical pathologist view, the opposite end to that we've just heard about in terms of trying to be at the other end of Google Earth, trying to understand the spatial geography of the pancreas in cystic fibrosis. So I'm going to briefly review clinical pathologist descriptions of how CF progresses in the pancreatic disease before telling you how we've designed semi-quantitative and now truly quantitative ways of measuring the processes and also distribution of specific cell types. We're setting up some in vitro models to try and move beyond snapshot cross-sections to elucidate how these signaling pathways between exocrine and endocrine pancreas may be working, and beginning to look at the changes in the endocrine compartment within the pancreas and in the models. Finally, very briefly, I can see in the audience there actually, I'm going to touch on some of the wonderful metabolic and imaging together with circulating biomarker studies that's happening in our collaborative group in Copenhagen. So cystic fibrosis described in 1938 was described around the appearance of the pancreas being cystic and fibrotic. And as you know, up to 90% of people with CF have pancreatic exocrine insufficiency. And CF-related diabetes is the most common comorbidity, affecting maybe 20% of adolescents, but as people are getting older, at least 40 to 50% of adults. And what's really striking is that if exocrine function is normal, insulin secretory capacity also remains normal. So I was really fortunate a few years ago now in being able to lead, funded by the CF Trust in the UK, something called a Strategic Research Centre in CFRD. And it's a really great way of funding, bringing together early career researchers and scientists who haven't previously worked in CF to get together and begin to face some of the challenges. And one of the first things we did, we've just been hearing about it in the previous talk, was to try and localize expression of CFTR. And we took a tissue-based approach. This is Michael White in my group, who stained normal human pancreas for chromogranin A to identify endocrine cells in the red, and then using RNA scope in situ hybridization, localizing CFTR expression. And as you can see, this was very much localized in the ducts and in fact was virtually absent from endocrine cells. And Michael backed this up with validated antibody CFTR staining as well. At the end of that first consortium, which is entirely preclinical, we applied for further funding to move slowly towards the translational space and to think very specifically about the mechanisms in the exocrine pancreas which may be signaling rather than directly damaging beta cells in diabetes. This brought together my own team, Exeter, as you'll come to hear, Copenhagen, Lund, Munich, Budapest, Sydney, and here in Philadelphia with Mike Rickles' team and the next speaker's team who is sitting in the audience. The first thing we were able to access through the seminal pathologist working in this space, Günter Kleppel, was 11 historic donors who had died with CF. And we have a standardized suite of staining of hematoxin and eosin, serious red and fast green, with immunohistochemistry staining for islets, endocrine cells, and CD31 for endothelial cells, as well as inflammatory cells with CD45, and then dual chromogenic stains with insulin and pancreatic polypeptide, and glucagon and somatostatin. And as I said, we wanted to take an unbiased, pathologist-driven approach to these tissues. And Yvonne Berry, working in Newcastle, began to look at the phases of disease. And as you can see on the left, in early disease, there are blocked ducts that become increasingly dilated. On the right, this begins to be associated with loss of acinar tissue, and then replacement through fibrosis, and ultimately replacement of the acini with adipocytes. But as you can see, the islets remain remarkably intact within this highly damaged tissue. And here you can see how the whole pancreas becomes replaced with adipocytes, and yet, particularly on the left, you can see islets just with little strands of remaining collagen around them. So Yara Al-Selwi, working in my group, started categorizing these donors into the control pancreas patients, those with fibrosis alone, those with lipoatrophy alone, and those that had a mixed picture, as you can see in the middle there, of both pathologies in one block. And I think this becomes very important when you want to do deeper analysis to look at the range of pathology within single blocks. And all of the younger patients in the series had purely a fibrotic pattern, and the older patients had either lipoatrophy alone, fibrosis alone, or a combination. And you can see that Yara separated them into the blocks with 7 controls, 7 fibrosis alone, 8 mixed, and 10 lipoatrophy. And Professor Kleppel has previously published semi-quantitative analysis of the different compartments and the changes within those. The lumen dilatation, fibrosis, loss of ducts, inflammation, acinar atrophy, and then those anatomical changes that take place within the islets themselves. So there's a complicated lot of data to look at here, but you can see moving across the graphs from normal, fibrosis, mixed picture, and then fat replacement. And what strikes me is that you see increasing duct dilatation very much mirrored by fibrosis. But then in the end-stage disease, not only do you get fat replacement and loss of ducts, but largely loss of the fibrotic tissue. Inflammatory infiltration seems to increase throughout the phases of the disease, including in the end-stage disease. You can see how acinar atrophy ensues increasingly as the disease progresses, but also how the islet shape and atrophy becomes increasingly worse with disease progression. So we were then really fortunate to work with Sarah Richardson, who's a wonderful scientist in Exeter. Many of you will know her work here through NPOD, and we were able to pool resources. So they have a fantastic archival diabetes bank, but also which contains many control young donors and extra donors with cystic fibrosis. The CLEPL series, NPOD provided access to donors without CF, and we in Newcastle have now collected a large number of organs, again, without known diabetic disease. Sarah and the team scanned in the slides that we'd stained in our quality assured clinical lab in Newcastle using Vectra, and then using Halo software was able to classify individual regions and cell types for quantitative representation and analysis. So here you can see, here's Sarah with her team of wonderful early career researchers, and in the blue we have tracked by age the number of donors without CF. As I said, really important, the controls showing without CF, and it's really important that we've got young donors in there, and then the CF donors in blue, my apologies. So let's now look at these quantitative graphs. So here, the controls in red, acinar volume making up virtually 100% of the pancreas, not changing from very, very young to older donors. But look at the beginning of that graph, and you can see that in cystic fibrosis at birth, acinar percentage area can be down to as low as 50%. And then you see a rapid progression and loss of acini until there's no acini left after the age of around 10. And you can see how beautifully that's mirrored by the replacement with fat, with virtually no fat in the donors without cystic fibrosis, and then this increasing fat replacement up to 60 or even 80% of the tissue blocks. So let's look at the last figure here, looking at endocrine. So this is looking at islet area as a proportion of the whole tissue. And this is where the age becomes really important, because it looks like endocrine area is decreasing with time. But of course what's happening is the rest of the pancreas is enlarging more quickly, so that the percentage endocrine area appears to fall. And it very much is mirrored between normal young donors and CF. The next thing we were able to do is to look beyond just serous red fast green as a measurement of collagen density and in fact intensity in the whole block to see what's happening around the islets themselves. So by drawing a halo 30 micron, you could probably just see it in the light blue beyond the outer edge of the islets. Matt Palmer, working in Sarah's group, was able to quantify peri-islet collagen density. And you can see just how much greater this is than in the normal donors. And that doesn't exclude those who have fat replacement. I think it's a very important finding. So Yara in my team is going back to that qualitative approach and beginning to look at other cell types. So in the middle panels, you can see serous red staining, fibrosis increasing and then becoming less marked in lipoatrophy. The bottom panels show inflammatory cell infiltration, but Yara is particularly interested in looking at activated fibroblast, pancreatic stellate cells expressing alpha-smooth muscle actin. And she's been seeing how this increases in the early stages of the disease, mirrors serous red staining of collagen deposition, but then appears to become relatively quiescent at the end stage. So this is a donor without CF, but a pancreatectomy donor, where she's beginning to see the collagen distribution. And if you look at the right with the alpha SMA staining, you can see it nicely localized around the ducts and then localized around the islet above the duct. So Yara hypothesizes that ductal dilatation may lead to activation of these cells. We've just been hearing about that. And then we were asking what cells are being activated. And she wants to explore, is this the fibroblasts, which then drive excess collagen deposition? And is that fibroblast activation and fibrosis signaling changes in the endocrine cells leading to dysfunction? Because residual islets are still there. They're just not working in CFRD. And this very much mirrors, we were really lucky in having John Engelhardt in our first consortium, a wonderful, wonderful scientist, as you know much better than I do. And he was analyzing ductal secretome in the fantastic CF ferret knockout model and implicated TGF-beta signaling, Wnt, Notch pathways as potential upstream regulators. And the downstream targets postulated to be including activated fibroblasts through upregulation of COL1A1 to deposit collagen, but also this candidate marker IGF-BP7. And in the figure from his paper, you can see bottom left, IGF-BP7 is usually expressed in the ducts. But on the right, in CF, in ferrets, IGF-BP7, this is by in situ hybridization, is expressed in fibroblasts. Further to the right, you can see isolated ferret islets treated with low glucose LG and high glucose, seeing a nice glucose-stimulated insulin secretion. Incubation with the candidate protein IGF-BP7 leads to higher basal secretion at low glucose and loss of glucose-stimulated insulin secretion in ferrets. So we went back to our human tissue and thought, how can we begin to study these cells ex vivo? Took human islets from normal donors, and Annalisa Flatt, now more recently working here in Philadelphia, and Nicole Katner, grew islets in 2D, allowing fibroblasts to grow out of them. And you can see in the green staining, vimentin staining of fibroblasts, where insulin and glucagon staining is being lost over a very short time period. So here you can see Yara's developed this model, day 0, 1, 3, 5, and 7 days. And she showed that these fibroblasts are indeed activated with alpha SMA expression and the number of cells expressing alpha SMA growing out and proliferating over time. And of course, she looked for IGF-BP7 and could see that this was highly expressed at a protein level by IF staining in these cells with increased number of cells expressing IGF-BP7 over this short 7-day model time course. We then worked with Anna Gloin and particularly Nicole Krentz, who did bulk RNA sequencing on samples taken. You can see all of the samples mapped onto these graphs at starting point, day 3, day 5, and day 7. And just how dynamic this system is, with ductal expression, keratin 19, increasing at day 5 and then falling away a little bit, whereas vimentin expression increases day by day. Alpha SMA is highly upregulated at day 7, and these cells are activated in terms of expressing the gene COL1A1. IGF-BP7 is indeed upregulated in these human mixed cultures, but with a preponderance of fibroblasts. So Caitlin Brack in my group started looking at secretome, and collagen is indeed expressed and secreted by these cellular models, but also IGF-BP7 protein, as you can see here. And recently at the EASD, Ephraim Vestholm and Lena Eliasson presented their data using IGF-BP7 on human beta cell lines and reflecting the ferret data, had a decreased glucose-stimulated insulin secretion by very short incubation with IGF-BP7 of human beta cells, but longer incubation leading to a phenotypic shift with changes in gene expression and protein expression. So that's one candidate gene. Nicole has found a few more, as you can see from here. How many genes are commonly upregulated as this model develops in vitro, with more than 1,000 upregulated genes shared between the different time points compared to baseline, and more than 1,000 downregulated genes. And the pathway analysis that Nicole's beginning to undertake is indeed showing downregulation of pancreatic exocrine secretory pathways and endocrine secretory pathways, with an increase in cell cycle pathways, hippo pathways, but very much extracellular matrix. So we now want to begin to look further at the impact of secretome from these human cells on rodent and human beta cell lines, both in terms of glucose-stimulated insulin secretion and indeed the phenotype of the cells. And once we can see a functional impact, then we can begin to do a much deeper genomic, transcriptomic, and protein analysis of the pathways. We want to see whether co-culture of activated fibroblasts is driving this, very much as we heard earlier on. We want to see whether secreted exosomes may be mediating some of this signaling between the exocrine and endocrine compartment. And then we can start beginning to break down and individually over time test candidate signaling molecules. Then, of course, we return to the human pancreatic CF tissue for a deeper spatial analysis informed by the in vitro studies. So coming towards the end, what have we learned so far around islets? Well, as I mentioned before, we do see not a huge change in terms of density, but what we see is the fact that the average area becomes much larger through clustering, and that there's a small decrease in the percentage of area compared to normal controls that's occupied by islets, but it's relatively small. And indeed, the hormonal staining that I talked about earlier is showing that in early disease and indeed in late disease, insulin expression is still very prominent. That's the brown expression on the left, with PP expression shown in red. And on the right, glucagon expression also prominent and very much maintained in the most end-stage disease. I mean, it's quite incredible, really, to see this hormonal expression surrounded by fatty replaced tissue. So Nicole Kattner has begun to semi-quantitatively, indeed quantitatively, but manually count these phenotypes. And of course, we're now working with Sarah for automated image analysis of maintained insulin and glucagon expression in all of the blocks. So a few years ago, Michael White was the first to publish the possibility that insulin-positive cells can also express glucagon in type 2 diabetes as a simple way of looking at transdifferentiation in human tissue. And also, interestingly, that co-expression with vimentin was often seen. So if you look at the bottom panels and the arrows, you can see green-stained endocrine phenotype cells co-expressing insulin in purple. So we went looking for these cells in cystic fibrosis, and here's an early donor with fibrosis alone showing vimentin staining, as you would expect in the fibroblasts and perhaps pericytes, with glucagon staining in purple and insulin staining in red, and no co-localization. And when we go to more advanced disease, we indeed don't see insulin co-expressing glucagon or vimentin, but we very, very much see glucagon cells expressing vimentin. So I think it's really striking how endocrine-like these cells look that are expressing vimentin in the middle of that islet, and how that they're expressing glucagon in parallel. So Nicole has, again, manually counted islets that have fibrosis around them and islets that don't have fibrosis around them to look to see whether the fibrosis may be associated with this phenotypic shift in the alpha cells. And there's a very striking relationship with an absence of this phenotype when there's no peri-islet collagen and a high preponderance, indeed, of many cells within each islet surrounded by fibrosis expressing this. And Nicole's working hard with Sarah now, and we've just seen the R-squared value of really nice correlations between AI-driven fibrosis peri-islet measurements and vimentin glucagon co-expression. And we're now working with two teams in Stanford to begin to look by single-cell RNA-seq at glucagon cells with and without vimentin. So just in my last slide is just really an advert for Bibi Nielsen and Daniel Farholt Jepsen, who've been doing incredible clinical studies in parallel with more than 100 extended oral glucose tolerance tests before starting triple modulator and now a year on triple modulator. And I won't go through these data, they're unpublished, but they're showing how you can begin to look at changes in insulin secretion in parallel with changes in proinsulin and glucagon secretion to begin to try and interrogate that intra-islet hypothesis in patients. And exosome samples are being collected, microRNA samples, in addition to high-intensity imaging to look for MR changes. So, to conclude, cystic fibrosis is characterized by progressive fibrosis of the pancreas, but ultimately lipoatrophy, but the residual islets are still surrounded by collagen. Disease progression associated with fibroblast activation, we're exploring through in vitro modeling using human-activated fibroblasts and trying to elucidate the impact on endocrine cells in vitro. There's clearly a high proportion of beta cells, even in absolute end-stage cystic fibrosis, and we're beginning to look at correlations with altered phenotypes, and actually, interestingly, alpha cell phenotypes with those changes. The beauty of the Strategic Research Center is that we're working with clinical cohorts to undertake parallel metabolic and imaging and biomarker studies so that we can begin to understand mechanism when we don't have tissue samples. Thank you very much. It's been a massive, massive team effort, as you can see, many countries, many fantastic scientists. This is my team. This is celebrating 100 pancreas organs collected into the tissue bank. And it allows me to say, it was a wonderful celebration, but we are so thankful to the donors and their families for providing this tissue. Otherwise, we wouldn't be able to do the work. Thank you.
Thanks, Jim. That's great. I have one question in the iPad from Michael Narkowitz. Sorry if I said that terribly. Yeah, I was going to say, okay, so he answered your question a little bit, but maybe you want to expand on it. He asked, how might the tissue-based data that you've collected translate to more clinical assays, for example, in blood, to advance risk stratification for individual patients?
Yes, indeed. So working with Shafak Waters in Australia, she's got really good projects going on looking particularly at exosomes collected from patients and trying to understand the correlation between exosomal microRNA and protein complement and lung disease, but also potentially progression to pancreatic endocrine dysfunction. We hope that we'll be able to demonstrate whether those exosomes have been produced by exocrine or endocrine pancreas through collecting them in our model systems and potentially in patients, because I couldn't agree more. I think we need to combine imaging and circulating biomarkers to understand what's happening at a tissue level, and indeed to look at disease-modifying impacts on tissue, we need meaningful biomarkers. So we're going for exosomes, but also microRNA. I think cell-free DNA is also interesting, but we're coming back up from the tissue.
Yeah, I agree with that. Maybe a follow-up question, but actually also sort of a different take. Same question as asker. Does the data on acinar cell loss over time that you showed, that is virtually all gone by 8 years of age, suggest that there might be a threshold age for improvement or preservation of exocrine function with the current therapies?
Yeah, I think that's a great question. And I haven't really thought about it from the exocrine point of view, although I have, because obviously I think that maintaining exocrine mass and function and non-scarring signaling is really key. It definitely makes me think about this, and it's something we've talked about with the Copenhagen team. You know, why is it that the damage is occurring so early and seems to be so complete, and yet the islets may have completely normal function or very much failed function? So I think all of these data suggest that there are windows of treatment while the pancreas is still developing, as soon as possible, maybe there is a threshold around about 8, 9, or 10. I think in pancreas disease generally, we believe that the pancreas, people talk about proliferation and can we reinduce beta cell self-duplication. And my feeling is very much maybe up to the age of 20 when the pancreas is still growing. But then I think you have your end-stage beta cell mass, and then I think we're talking about function, whether that's pregnancy, obesity, scarring, pancreas disease. But that's a bias rather than a fact at the moment.
Okay, great. Thanks so much, Jim. Okay, let's move on. Okay. It's my pleasure to introduce our next speaker, Andy Kelly, who's hosting us here in Philadelphia this week. Andy's an attending physician at the Division of Endocrinology and Diabetes at Children's Hospital of Philadelphia and professor of pediatrics at the Pillman School of Medicine, also in Philadelphia. She's going to talk to us about the effect of modulator therapy on pancreatic endocrine function. I'm going to set you up. Okay. Or you can just hit the arrows now. And then 1 second.
I was just telling Jim last night at dinner I had asked him to save me some minutes because I was close to 30 and he did not. So I'm going to probably skip over some slides like Dr. Galton. Okay. But I could not think about the symposium title, Pancreas on a Pedestal, without acknowledging the Mutter Museum here in Philadelphia, which is a really wonderful medical museum. And so if you all have the opportunity to see it while you're visiting Philadelphia, I'd highly recommend a visit. That's the only advertisement for this meeting, or for my session. So today I'm going to review some basic principles of glucose excursion and islet function in people with CF. And I've been tasked with talking about endocrine pancreas, but I want to acknowledge that I will largely be speaking about insulin secretion. And like Dr. Shaw has mentioned, we are getting things like glucagon, incretins, but those aren't ready for me to present today. I'll review some of the currently available literature and emerging data on the relationships of modulator therapy with glucose tolerance and insulin secretion, but this will be patient-oriented. I know that there's some literature both in vitro and in the animal models suggesting that there is clearly a role of modulator therapy in islet function. I will reserve that for the basic scientist. And I will interweave through this some potential mechanisms, patient characteristics, time frame that might confer benefit to improvements in insulin secretion. Because it's a mixed crowd, I thought I would just review some basic tenets of CF-related diabetes. Jim has already reviewed with you that a good 40% of individuals age 30 and older have CF-related diabetes. We see initial appearance, usually in later childhood, but we see that by OGTT, and recently my colleague Mike Stalvey showed me yesterday some data in using continuous glucose monitoring in toddlers, frequent glucose excursions to greater than 200. So we're seeing hyperglycemia pretty early on. Usually the transition is subtle from some minimal glucose abnormalities and over time worsening of those glucose abnormalities to frank CF-related diabetes. But every once in a while we do have particularly young patients who quickly do not have that, who don't have that prodrome and quickly have pretty significant CF-related diabetes. And a subset will have fasting hyperglycemia. So I was thinking about Dr. Shaw's data and how those pancreases might look. Fasting glucose tends to increase over time, but frank hyperglycemia tends to happen much later in the course of CF-related diabetes. And as many of you know, or most of you know, the diabetes occurs largely from insulin secretion defects. I'm going to take a fair amount of time to review this slide because it has been used to inform the PROMISE study data that I will share with you today. In addition, it will also speak to the challenges that we have in trying to figure out how best to understand the data. Here I'm presenting mixed meal tolerance test data. The data will look similar if this were an oral glucose tolerance test. And these are data that I've had the great fortune of presenting today through a close collaboration with my colleague Mike Rickles at Penn. So on the left-handed graph, I'm showing plasma glucose. In gray is is glucose tolerance in an otherwise healthy individual without CF. You can see that glucose tends to peak by about 30 minutes and tends to be lower than baseline by about 2 hours. And the reason for this glucose profile is the insulin secretion shown here on the right, and courtesy of my colleague Darko Stefanovsky, also at Penn. And what you can hopefully appreciate is in that first 30 minutes of a meal or a glucose tolerance test, there's really robust insulin secretion. And we often refer to this as first early phase insulin secretion, and is likely equivalent to the first phase insulin secretion that one might see if you gave an IV bolus of glucose. This is accompanied by a less robust second phase insulin secretion with insulin peaking at about 45 to 60 minutes. Here in green are individuals that we refer to as having normal glucose tolerance. And that is a glucose that's completely normal at 2 hours and lower than 150, 155 at 1 hour. But I would submit to you that while we're referring to that as quote unquote normal, that 1 hour glucose is higher than what one would expect in an otherwise healthy individual. And here on the right, perhaps some dampening of that early phase insulin secretion, but then a really robust sort of later second phase insulin secretion likely to compensate for that relative hyperglycemia at 60 minutes. In purple is what we refer to as early glucose tolerance, so someone who might be considered to have subtle glucose abnormalities, an isolated glucose at 1 hour greater than 155. And here you can see that you have further loss of that first phase insulin secretion accompanied by a right shifted and dampened second phase. In orange and red, impaired glucose tolerance and CFRD with further loss of that early phase insulin secretion and a further delayed and further dampened second phase insulin secretion. And I also would like to acknowledge that late insulin secretion is likely responsible for the frequent hypoglycemia that we see 3 to 4 hours after a glucose tolerance or after a mixed meal. So, some modulator therapy data. Many of us who were caring for patients with CF noted after the initiation of ivacaftor that patients were requiring less insulin or were having the sudden appearance of hypoglycemia, and this motivated a number of studies. Here I'm showing data from Melina Bellin, who looked at 5 individuals who had been treated with ivacaftor for just a month or so. She performed oral glucose tolerance tests as well as acute insulin responses to glucose and demonstrated in that in individuals with normal glucose tolerance and this one person with impaired glucose tolerance that perhaps there's some improvement in insulin secretion under the oral and IV conditions. The results in individuals with CFRD were certainly less robust, if present at all. And this suggested to me, at least in the short term, that if you're going to see an improvement in insulin secretion, there needs to be some minimal residual beta cell function for that to happen. I think more compelling are the registry data from both the US and the UK demonstrating trends in lower CFRD rates over the 4 to 5 years after the initiation of ivacaftor therapy. And this suggests to me that it may be years before we see any delays in progression to CFRD, and perhaps maybe we'll see some prevention. What we can't glean from this data is if there's any reversal of CFRD. Amir Mohit examined oral glucose tolerance test in 39 individuals who were enrolled in the the CF Foundation Prospect study that examined multiple outcomes in response to clinically prescribed lumacaftor or ivacaftor in people who are homozygous for delta F508 mutations. And hopefully what you can appreciate, at baseline there was a fairly variable glucose tolerance, but no clear improvements in glucose tolerance over that 12 months. Here are the data shown more granularly, no improvements in fasting glucose, 2-hour glucose or glucose area under the curve, and no improvements in insulin area under the curve. Many of us, however, weren't surprised because we recognized that this lumacaftor-ivacaftor therapy was less robust than ivacaftor for the gating mutations with respect to primary CF outcomes like sweat chloride and FEV1. Amir went and looked at these data a little bit differently and said, let me look at just the subset of individuals who have relatively preserved insulin secretion, i.e. indeterminate or impaired glucose tolerance. He had 10 individuals at follow-up, reduced from that 15, and suggested that perhaps there were a subset of individuals who had improvement in their glucose tolerance from the small stub study. There's a similar study by the Miskolc group in Europe in whom individuals with either impaired glucose tolerance or CFRD that was newly diagnosed at baseline were followed for a year and had repeat OGTTs at 12 months following the initiation of lumacaftor-ivacaftor. And their data also suggested that perhaps there was improvement in their glucose tolerance. And I meant to go back to last night. I'm not sure that they differentiated between normal glucose tolerance and indeterminate glucose tolerance or just relied on those 2-hour glucoses. And then more recently, Kevin Scully looked at continuous glucose monitoring in individuals with established CF-related diabetes prior to and a few weeks following the initiation of elexacaftor, tezacaftor, ivacaftor, which I will now refer to as ETI, and also demonstrated improvements in glucose excursion. And here's an example for individuals who are unfamiliar with continuous glucose monitoring. You can see the multiple glucoses over a 24-hour period overlaid with multiple days overlaid, and hopefully it's quite clear when someone ingested a meal. This is data from Annalisa Flatt, whom Dr. Shaw allowed us to work with for about 10 months, and she continues to work with us, and I would recommend that you see her poster. She was largely working in the islet field, but did some secondary data analysis on some data that we've been collecting for many years on individuals with CF. And she's looking at glucose potentiated arginine stimulation test and mixed meal tolerance test. I will just, for the sake of this talk, present some of the mixed meal tolerance test data and recommend that you see Annalisa's poster. So what she did was look at the subset of individuals who were for whom we had mixed meal tolerance tests prior to and following ETI. And then what she did was take a similar group who was not treated with ETI. And there's a couple things that I want you to observe from these data is that the time between individuals baseline and follow-up mixed meal tolerance tests was much longer in the ETI group versus the control group. Here are their glucose tolerance test data on the right, those controls not treated with ETI. And while many of them started off with normal glucose tolerance, again, small group, they seem to have a worsening of their glucose tolerance. In contrast, the individuals who were treated with ETI seem to have perhaps improvements or less worsening over that time frame. Now, that may have been their natural history. It's really hard to glean, but there is some additional data here from their mixed meal tolerance test suggesting that there may be something happening. So on the upper panels is glucose. You see that the subgroup treated with ETI had lower glucoses compared to controls who really had no change. And then also perhaps this restoration of the insulin secretion profile with perhaps a shift in earlier insulin secretion, but certainly not normal because we want to see that maximum insulin secretion within the first 30 minutes. We did not see that in the control group, and perhaps a dampening of their insulin secretion is measured by C-peptide here. And then the WashU group conducted a retrospective study of 134 adults treated with ETI for about 12 months. Not unexpectedly, they had increases in weight and BMI, and increasing rates of overweight and obesity, as well as worsening rates of hypertension. What I want to call out here are accompanying those. In the subset, they had 20 or so individuals with CFRD for whom they had fasting lipids, and they saw increases in LDL and HDL, but no frank emergence of dyslipidemia. But what they did see is in the subgroup without diabetes, a lowering of their hemoglobin A1C. So promising data, which now leaves me to PROMISE. So for many of you in the audience, I imagine that you're familiar with PROMISE, which is a CF Foundation prospective study to look at multiple outcomes following the initiation clinically of ETI. And this study was started in 2019 following FDA approval. And Mike Stalvey and I, for the endocrine sub-study, are looking at individuals age 12 and older. And as part of that study, we are collecting multi-sample OGTTs at 0, 1 year, 2 year, and now out to 42 and 54 months. Some of the time frame is off because there was a 6-month delay related to COVID. And we're also getting DEXA for body composition and bone. What I want to highlight here is that we're looking at not only at glucose tolerance, but we're also looking hopefully with an increased granularity at the OGTTs by looking at glucose excursion, early phase insulin secretion, and adjusting that for glucose area under the curve. And I showed you those initial mixed meal tolerance test data because they're complicated data and we're still struggling or grappling with how best to address these things. And you can see we're getting glucagon and incretin as well as a number of other outcomes. I want to acknowledge that there's no improvement in fecal elastase thinking about this. Exocrine, endocrine, pancreatic potential intersection. Here are the baseline characteristics, which I won't go through. I also want to have a shout out to Mike Narkowitz. We do have 16 people who had OGTT and liver elastography, so I think that while it's a small group, it'll be nice to see if there's any sort of links between what we're seeing with glucose excursion and liver stiffness or perhaps fatty liver. Okay, these are complicated slides, so I'll try. I'm not going to walk through all of the details here, but I thought it was worth sort of walking through these sort of to sort of show you some of the challenges of looking at the glucose tolerance. We have approximately 65 to 67 OGTT at baseline for PROMISE. Here I am categorizing people by the classic CF Foundation guidelines, which include indeterminate glucose with a 1 hour glucose greater than 200. It was a little bit different than our threshold for early glucose intolerance, which is greater than 155. Hopefully what you can appreciate is that if you have normal glucose tolerance, you could remain the same or get worse over that year and a half. If you have indeterminate, you could stay the same or get better. And if you have impaired glucose tolerance, similarly, you could stay the same or get better, and CFRD similarly. So a big mishmash. I'm going to call out specifically one of the patients with CFRD in a later graph. Obviously, this was not a controlled trial, so then we went back to look at some of the OGTT data that we've been collecting from a research standpoint to try and sort of anchor these data. And what I wanted to show you that similarly, people in our cohort not treated with ETIs, these were all before 2019, have glucose tolerance tests that are all over the place at 12 to 18 months. Looking at this a little bit differently, we have OGTTs with glucose, just the glucose data available for 24 months in about 25 individuals from PROMISE. Here I'm using that slightly restricted glucose tolerance by defining it as early glucose intolerance. Again, a mishmash of what's going on, not any clear, I think, indication that glucose tolerance by our strict glucose categories is getting better, and this is similar to what we see in our CHOP-HUP cohort. Here, following up on that data further, just to show you the actual data, or box plots showing the fasting 30-minute, 60, and 120-minute glucose from that OGTT. The only thing that was different was the fasting glucose was slightly lower. I'm going to, in a few slides, show you more about that. Then we looked at, as I told you, we were particularly interested in, are we having some restoration of that early insulin secretion? Here's the first incremental area under the curve for glucose, not different. For those insulin secretory rates, not different. And then when you adjust the insulin secretory rate for glucose, not different. So we're not seeing in this broad group of individuals with normal glucose tolerance out to CFRD, on average, improvements in insulin secretion. No differences in insulin sensitivity either. Here are the insulin secretory rates, again, courtesy of my colleague, Darko Stefanovsky, not seeing any differences there when we do that by incremental area under the curve. But what is, I think, really fascinating when you look at these data a little bit differently, in green are the insulin secretory rates prior to ETI, and then in orange after starting ETI. And you get the sense that maybe that there may a subtle shift in improved insulin secretory rates in that first 30 minutes. Looking at the data a little bit more differently, peak glucose was also a little bit higher, a little bit sooner, suggesting perhaps some improvements in insulin secretion along the way. And this did not meet significance, but peak insulin secretory rate tend to be shifting over as well. But as you can see, it's late regardless. 85 to 78 minutes is much later than one would expect when you expect your peak insulin to be about 30 minutes. Looking at this again a little differently, so you can see how we're grappling with this. Insulin and C-peptide, I'm showing the C-peptide data area under the curve, and what we are seeing at 12 to 18 months is higher incremental area under the curve for C-peptide. And even if you adjust that for glucose. Oops, sorry, showing there. I wanted to go back to that fasting glucose because that to me was really intriguing. And what we see is that fasting glucose is lower, as hemoglobin A1C is also slightly lower. And we restrict it to exclude the 7 or so patients that were treated with insulin. Those lower fasting hemoglobin A1C and fasting glucose persist. So I wanted to look at that, is what's going on in the fasting state. Fasting insulin tended to be a little bit higher at 12 to 18 months, and C-peptide just reached significance. I'm not so sold on a basal insulin secretory rate because I'm not sure how good the model is, just with one time point. We didn't see any differences there, but more to come on those data. And then I wanted to just, I think, show these data a little bit differently, just give you some more granularity. Here in green are individuals with normal glucose tolerance, and their insulin secretion to glucose for the first 30 minutes. X-axis is pre-ETI and Y-axis post-ETI. So you can see there's a lot of variability in green. In blue, not unexpectedly, if you have early glucose intolerance, you're going to have lower insulin secretion for glucose. In orange, there's a lot of overlap between the insulin secretion for glucose. And then not unexpectedly in red, individuals with CFRD tend to have a lot lower insulin secretion. I want to highlight specifically this individual with CFRD who had really robust insulin secretion pre-ETI, lower insulin secretion for glucose post-ETI, but went from CFRD to impaired glucose tolerance. You can see why I'm struggling. Here's that individual, 27-year-old, good BMI, relatively good pulmonary function, clearly has CFRD by that OGTT, did become hypoglycemic. Here is his OGTT 12 months later. BMI is now 25, pulmonary function beautiful at 103. And what you can see is that he has impaired glucose tolerance, no longer CFRD, but you see that the glucose is higher in that first 30 minutes. So I went and paired this to the insulin secretion, and I want to point out that first 30 minutes, and you get the sense that there is some recovery of that early phase insulin secretion, but is accompanied by higher glucose. And so what you wind up with is a lower insulin secretory rate to glucose. And so I'm going to rely on Darko and Mike Rickles to sort of help me work through these complicated data. But you can see it's complicated, and but it does look like there is, at least in a subset of patients, improvements in insulin secretion, and you see that with the second phase as well. So I have no idea whether this is related to ETI, but I think it's interesting to speculate about the potential mechanisms. And we've heard today about potential direct and indirect CFTR effects on the islet or perhaps the islet neighborhood, which was where we think more likely. Could this be due to reductions in systemic inflammation, pancreatic inflammation, improved GI health? Bile acids play an important role in insulin secretion, or perhaps incretins. And then crosstalk between the liver and pancreas, I think, is worthwhile exploring. And then I think ultimately, as diabetes docs serving the CF population, are some of these positive effects, if they are present, going to be offset by the emergence of overweight and obesity-related insulin resistance, as well as not so great diet. I'm just going to follow up with, 2 slides, because this is a question that I often get. What's the relationship between hemoglobin A1C and BMI-Z? In blue, baseline, and in red are the follow-up. We did not see that BMI-Z conferred higher hemoglobin A1C in this group at 12 to 18 months. Then I also want to say, well, the hemoglobin A1C is better. Maybe it's completely unrelated to the OGTT, or we know that hemoglobin A1C and OGTT are often disconnected, at least that 2-hour glucose. So again, in blue are prior to ETI, hemoglobin A1C on the x-axis, on the y-axis is the glucose incremental area under the curve for the 2 hours, and then in red are the follow-up. And it looks like the relationship between these two are similar. So I don't think that hemoglobin A1C lower has to do with a different that the way the hemoglobin A1C captures the glucose any better. So I think I'm over time, or almost. Oh, I'm pretty good here. So at 12 months, evidence for improvement in glucose tolerance is lacking for ETI. There's some subtle improvements in hemoglobin A1C and fasting glucose that do not appear to be related to changes in BMI nor pulmonary function. I did not show those data. But there's this early suggestion of a shift to earlier insulin secretion, perhaps restoration of insulin secretion, at least in a subset of individuals. But who that subset are and what confers that positive benefit, I think it remains unclear. We are collecting the 24-month data, and I think that this will be important to solidify whether we are seeing improvements in hemoglobin A1C, fasting glucose, and perhaps even further restoration of insulin secretion. And you now know, and many of us know in the audience, that 12 to 24 months really is insufficient to see what's happening with glucose tolerance. And so the CF Foundation is funding us to go out to 54 months. And then I think it'll be really important for us to consider the intersections of improved inflammation, insulin resistance, liver disease, and a myriad of things that could be intersecting with the endocrine pancreas. So at that, I, again, like Dr. Shaw, wanted to thank our patients and their families, Steve Rowe and Dave Nichols for allowing me to participate, and now my co-PI Marty Solomon, Mike Stalvey, thank you, and Mike Rickles, Darko, Rachel, Heather, so many people, thank you.
Thank you, Andy. Time for a few questions. Let's see. Here's one. Do you think, Catherine Coutney, I should have brought my reading glasses. Do you think that there is any impact on patient nervousness, in quotes, on research OGTTs from adrenaline or cortisol? Could this impact differences between A1C and OGTTs? Stress responses.
It's a great question. I will tell you that the protocol we use is to place the IV, wait 20 minutes, and then get the first -10 and 0 sample, and then we average that -10 and 0 sample, and we look closely to make sure that those two baselines are similar. I think it is a potential issue. I don't know that this has impacted the clinical OGTTs that I've seen, but I think this is one of the ways that we've tried to get around some of this in the research setting. Yeah. Thanks, Catherine.
Oh, sorry. Thank you. Let's make sure they're all for Andy. Because I've only been looking at the Q&As.
I don't want any of Kyle's questions.
Okay. Clinically and anecdotally, you're able to decrease insulin in patients with CFRD on ETI or enzymes. I think maybe you got at that during your talk. I mean, are you able to decrease insulin in patients with CFRD on therapy?
I think many of us will say we've had mixed results. Some patients have been able to go down on their insulin, some no change, and some have actually had to increase their insulin. And many of us have speculated whether that's to improvements in GI health and absorption, and some of our patients are just eating better, but it's really been a mixed bag.
And do you anticipate continued changes in glucose tolerance over time on triple therapy? What do you anticipate seeing there?
I think that's why we're doing this study. I think our hope was that we would see improvement. We're certainly not seeing that preliminarily and definitively at 12 months. I think that this is a study that really needs to go out 4 to 5 years, sort of to the, similar to what was seen with registry, to actually see if we are seeing improvements. I have a sense that it may be not so much our CFRD patients for whom we're seeing reversal, but perhaps some delay in the progression for our, perhaps stabilization for our patients with earlier glucose abnormalities.
Yep, okay, great. Thank you so
Thanks.
much. Okay. Our last speaker for this session is Vikesh Singh. He's a professor of medicine at Johns Hopkins. And there we go. Okay. And he's going to talk to us about the effect of CFTR modulators on exocrine pancreatic function, which perhaps we have not heard enough about so far this morning. Go ahead. Do your first slide, and then you should be able to use the arrow keys.
Great. I'd like to thank the organizers of the NACFC, as well as the moderators of the session, for the opportunity to speak to you today. I didn't know that exocrine function was such a hot topic. I've never seen this many people in a room for a topic of this nature, but I'll go with it. I'd also like to thank the CF Foundation for an opportunity to participate in the second Digest class, as that certainly stimulated a lot of my interest in CF and its association with pancreatology. I have some disclosures, but they're not relevant to this talk today. A quick overview. I'm going to talk a little bit about the mechanism of pancreatic disease and cystic fibrosis, really quickly touch base a little bit on the CFTR modulator therapy, try to impress upon everyone that exocrine pancreatic insufficiency is a syndrome, and then we're going to talk about the impact of CFTR modulator therapy on exocrine pancreatic function, in that it may restore exocrine function in younger PI-CF patients. And then those with restored function, there is an increased risk of developing acute pancreatitis, as we've seen in a couple studies. And finally, as far as this section is concerned, discuss a little bit about some of the studies that show that there's a reduced risk of recurrent acute pancreatitis in pancreas-sufficient CF patients with a history of prior acute pancreatitis. And lastly, and very quickly, I'll touch base on some reasons why CFTR modulator therapy does not restore exocrine function in all PI-CF patients. So the mechanism of pancreatic disease in CF is quite simple, at least from a very elementary level. Certainly we know acinar cells are making digestive enzymes, typically in zymogen form. They're not activated until they reach the duodenum. But they've got to get flushed out of the acinar, out of the ductal area, once they're excreted into the ductal area, by the bicarbonate-rich fluid that is produced by the ductal cells. And of course, we know CFTR is a central molecule in that. It's also important to remember that CFTR protein, in fact, the place where it's most expressed in the human body is the pancreas. And its pivotal role is to make a very bicarbonate-rich fluid, and that's what you see on the right-hand side. So as opposed to chloride being more the central permeable molecule in other areas affected in CF disease, bicarbonate is really central to the pancreas. CFTR modulator therapy is certainly a thing I'm least of an expert on, and certainly there are a lot of people in this room with a lot deeper expertise than myself, but certainly we know there are many classes, 5 different classes of treatments that are either already available or are in development, including potentiators and correctors. I think we're all most familiar with those. And of course, there's a lot of work being done on the development of stabilizers, read-through agents, and amplifiers. And I'm not going to go through all of this, but it's simple to say we know that this has done we've made a sea change of impact on our patients through these therapies by improving both the structure and functional aspects of CFTR protein in the treatment of cystic fibrosis-related disease manifestations. So it's important to impress upon everybody that exocrine pancreatic insufficiency is actually a syndrome, and it has 4 domains. It consists of nutritional intake, nutritional need, pancreatic digestive enzyme delivery to the small intestine, and certainly last but not least, intestinal adaptation. However, as clinicians, what we mostly focus on is the pancreatic digestive enzyme delivery to the intestine. So what we're most focused on is what is the pancreas making, and how can we test that through direct and indirect testing to figure out whether or not exocrine function is intact. But all of these other areas are potentially just as important. And what do I mean by that? So we know that when an individual consumes a meal, that there's going to be controlled release of nutrients from the stomach into the intestine. And remember that that's a very acidic chyme, right? The gastroparietal cells are making acid that mix with food as one of the first steps in digestion. And then there needs to be neutralization of those acidic gastric contents, right? And that's coming from the bicarbonate from the pancreas, right? So it's not just the digestive enzymes making their way into the intestinal lumen. It's also the presence of that bicarbonate-rich fluid that then neutralizes that acidic gastric chyme. So you have relatively a neutral pH throughout the rest of the small bowel to facilitate both the digestion and absorption of macromolecules. So remember that also we need not only pancreatic digestive enzymes, we also need bile. So we sometimes forget that patients with advanced cirrhosis who have limited bile production capacity or people who have impairment in their enterohepatic circulation can behave as if they have non-pancreatic exocrine pancreatic insufficiency, right? Or non-pancreatic exocrine insufficiency. And in fact, that's something else that we often forget, is that bile is also quite important. And finally, well, what about making sure that we have a sufficient luminal intestinal surface area, right? Because once you've digested something, you certainly need to have the surface areas that's going to allow for appropriate absorption of fat. I think the other thing I've not added to this, but I think there's a lot of growing data on, is the importance of the microbiome in exocrine pancreatic insufficiency. We do know that our CF patients are often on prolonged and changing courses of antibiotics, and that certainly does a job on their microbiome. And remember that when you have changes in your microbiome, you can also have small intestinal bacterial overgrowth, and that can also lead to non-pancreatic exocrine insufficiency. So now turning a little bit towards what have we learned in the last several years as to the impact of CFTR modulator therapy on exocrine function. Our first inkling that maybe something positive was happening was from this study. This is the ARRIVAL study, and everyone in this room is probably quite familiar with it. It was the first study evaluating ivacaftor treatment in patients with cystic fibrosis, children between 12 and less than 24 months. This study showed, and you can see nicely on the both upper and lower left-hand side, that the mean absolute change in fecal elastase levels actually went up about 165 micrograms per gram with a pretty reasonable confidence interval from baseline to 24 weeks of treatment on ivacaftor. This was then followed by the KIWI and CLIMP trials, which showed that elastase levels increased among CF children between the ages of 2 and 5 years with a CFTR gating mutation who were also treated with ivacaftor. So everyone the patients who started in KIWI then went 24 weeks of treatment, and then they were enrolled in CLIMB, and then that was an open label extension study that went out to 84 weeks. And as you can see here, the mean fecal elastase increased about 129, and of the 6% of patients had an increased fecal elastase over 200 at baseline, but at the end of 84 weeks, 35% of these patients actually had a fecal elastase over 200, suggesting that these patients had actually restored their exocrine function, at least on the basis of fecal elastase testing. More recently, my group and the group at Ohio State conducted a scoping review looking at the impact of CFTR modulators on the exocrine pancreas, and a scoping review is certainly a little bit different than a systematic review because it's not based on really high quality data. It's supposed to be something that drives hypothesis generation and those kinds of things. So this has a lot of case reports and case series, but if you think about it, that's largely what we have to date to guide our clinical practices in terms of how we think of CFTR modulators and their impact. But this scoping review evaluated 24 studies, 17 case series, and 7 case reports, 345 subjects. 85% of these patients had some assessment of their exocrine status at baseline and after CFTR modulator therapy, or at least while it was ongoing or during follow-up. 86% were pancreas insufficient, and 21% converted to pancreas sufficient CF. Now, this is based on, of course, 24 studies, 17 case series, 7 case reports, so about 20% there. Now, when you look at the individual level data, and that was only reported in 4 case series and 8 case reports from this, so a smaller subset of studies, which had a total of 34 subjects, all in the single digits with a mean age of 7 years. 32 had a fecal elastase less than 200, and after a median duration treatment of about 104 weeks, so that's about 2 years. 16, or 50%, had increased their fecal elastase to above 200 micrograms per gram. And one of the things that we had reported in this study was that the conversion from PI to PSCF versus those who remain PI was more common in those who had a detectable fecal elastase at baseline. That means they still have some acinar cell reserve. So that was 75% versus 0. So you need to have some acinar tissue left around. And those who are younger, 5.9 versus 8.4 years. Another study that had come out more recently, just published in fact this year, suggests that in those patients in whom you restore CFTR function, they could be at an increased risk of acute pancreatitis. And there were a total of 5 patients reported in this case series. And as you can see, they had a fairly consistent genotype, class 2, class 3 mutations. Almost all were treated with ivacaftor, with a couple treated with ivacaftor, lumacaftor combination therapy. Not all the studies reported fecal elastase at baseline and at follow-up, but among these 5 patients, one in fact had acute pancreatitis, I think 5 times. So clearly we are allowing some of that acinar cell reserve that's been sort of quiescent to wake up and cause pancreatitis without necessarily restoring their fecal elastase levels, because 3 out of 5 remain pancreas insufficient. The final point on the impact of CFTR modulator therapy for its impact on exocrine function are a couple of studies that have been published that suggest in those patients who are pancreas sufficient, who've had a history of acute pancreatitis, if you treat them with CFTR modulator therapy, they will not have any further bouts of acute pancreatitis. And the first study to suggest that was this case series, which Dr. Friedman was an author on. He's here in the audience. This was now reported back in 2018, so a good 4 or 5 years have passed. And other than one patient who had, I think, 1 or possibly 2 episodes in the first 3 months of therapy, everybody else up to 12 months had not a single episode of acute pancreatitis. We then also found something very similar when we looked at our series at Johns Hopkins in the adult cystic fibrosis clinic. On the top panel, part A, you can see there are a total of 15 patients with pancreas-sufficient cystic fibrosis, and each dot represents when along their course they've had an episode of acute pancreatitis. And that straight up and down arrow is when their CFTR modulator was initiated. And as you can see, over a mean follow-up of 3 years, there was not a single episode of acute pancreatitis among this group of 15 patients who were treated with the CFTR modulator and who had a history of acute pancreatitis. In contrast, in the bottom panel, part B, those are patients who also had pancreas-sufficient cystic fibrosis, but they had never had a CFTR modulator initiated. And while, of course, this was not a randomized trial, interestingly enough, you continue to see episodes that are occurring in those patients over a roughly similar time frame. The group from Ohio State did an administrative database study looking at CFTR modulator use and reporting that it actually might reduce pancreatitis hospitalizations in cystic fibrosis patients. Of course, we know administrative data can be difficult, right, in terms of how do you figure out somebody has pancreas insufficient CF versus pancreas sufficient CF, and we know even acute pancreatitis can sometimes be over-coded, but nonetheless, among the nearly 2,000 individuals in the market scan database who were treated with CFTR modulators, there was a 64% and 47% reduction in acute pancreatitis hospitalizations for those with PSCF and PICF respectively, so for what it's worth. But it looks like it's certainly doing something. If it's not necessarily preventing acute pancreatitis, it might be keeping people also out of the hospital because it's maybe reducing to some extent even pain that would not necessarily lead these patients to seek hospitalization. It's important to remember that there are potentially several reasons why some of these patients are not restoring their function entirely. So going back to the first point, we do know that there are many environmental factors associated with CFTR dysfunction. We know both alcohol and cigarette smoking interfere with CFTR function. And the one we also don't talk about much is how estrogen impacts the CFTR channel and function, leading to one hypothesis as to why women who are menstruating or are having active menses, in fact, have CF exacerbation. So we know that environmental factors are certainly associated with CFTR dysfunction. And let's remember that acute pancreatitis, like a lot of inflammatory disorders, is really at the end, is a complex interplay of both environmental and genetic factors for not only the initiation, but also the progression of this condition. So on the left-hand side, you have a whole host of environmental factors that are involved in the progression, but not only the development, but the progression of pancreatitis. And then you have a whole number of genetic factors. Initially there were 4, and now we're up to about 14 genetic factors that we know are implicated in the development of pancreatitis. Of course, CFTR is one. I sometimes talk about hypertriglyceridemia because this is also just a classic condition where we've learned a lot about how risk of or triglyceride levels are determined by a combination of both genetic risk and environment. And you can see here, obviously, about 72% of the population has a normal triglyceride level, under 177 milligrams per deciliter. But as you work your way out to the right, you can see that there are going to be individuals who have monogenic hypertriglyceridemia that only comprise about 0.1% of the population. But then you have a bunch of people in the middle who really have multigenic hypertriglyceridemia, which is really the combination of heterogeneous mutations of large effect and the cumulative effect of small effect variants that drive the risk of hypertriglyceridemia. And that's very different than these monogenic patients who may have homozygous or compound heterozygous for large effect loss of function mutations. And on top of that, you've got all the environmental factors that can sort of be in place that can also contribute to increasing triglyceride levels, many of which we all know in this room: obesity, diet, increased alcohol consumption, diabetes, renal disease, lupus, steroid use, oral estrogen, and so forth. Another reason might be that mild differences in genotype among CF patients can result in marked differences in phenotype. And this study struck me. This was Tanya Gonska's group. They published this paper about 3 years ago. And they looked at a cohort of individuals who carry the R117H mutation in combination with either a 5T, 7T allele. And what's curious is if you look at the far left-hand side, you've got patients who have, everybody here has, the first two groups on the left-hand side have cystic fibrosis. And you can see the only thing that differentiates them is actually the presence of either a 5 or 7T allele. And on the far right, or the last box at the very bottom that has patients with a normal sweat chloride, those are patients who have CFTR-related disease. So these are typically patients who have mild CF mutations, heterozygous, usually class 4, class 5, who can develop pancreatitis. They can even develop congenital bilateral absence of the vas deferens, but they typically don't develop the sinopulmonary problems that patients with classic CF develop. But you can also see that while there's not a difference in the rates of pancreatitis, between the 5T and 7T groups, so if you look at them compared to the CFTR-RD groups, there is a difference. And the only reason I put in on the little schematic on the right-hand side is just to show that the intron for exon 9, for the CFTR gene, is where there is a polymorphic site, and that polymorphic site can actually, it can have either the presence of 5 thymidines, 7 thymidines, or 9 thymidines. But the problem is when it has the 5 thymidines, you're more likely to skip exon 9, and if you skip exon 9, then you're not going to make enough CFTR protein, whereas if you're able to incorporate it, then you'll have a normal CFTR protein. So I think it's important just to sometimes go back and think about that, because just that small difference can make a really big difference in terms of where these patients phenotypically end up. Let's also remember that mutations in genes that affect trypsin activation and pancreatic secretion are enriched in CF patients with pancreatitis. This was a very nice study now published about 4 years ago. And if you look at the top group there, those patients with CF who have a history of recurrent, acute, and chronic pancreatitis, 48 of them, you can see that 29% of them had an intrapancreatic activation of trypsin gene. 23% of them had a pancreatic secretory pathway mutation, and a total of almost 40% had both. So, and when you compare them to patients with CF without pancreatitis, you can see that there is a statistically significant difference across all those groups. And why is that important? I mean, this could actually lead to underlying subclinical pancreatitis and maybe even fibrosis, as we do know that there are differences in the chronic pancreatitis population based on the presence of SPINK1 and PRSS1 mutations. For example, this study, which is now almost 20 years old, suggests that patients with SPINK1 mutations more rapidly progress to calcifications, which is the hallmark feature of chronic pancreatitis. They also more rapidly progress to both diabetes and exocrine insufficiency compared to patients with PRSS1 gene mutations. So can you imagine if you have CF and one has a PRSS1 gene mutation, another CF patient has a SPINK1 mutation, maybe they're going to behave a little differently. Their pancreas might look quite different under a microscope, and that would sort of be my next point, is we've also come to learn in patients with chronic pancreatitis that the histopathology of the pancreas actually looks different in patients with SPINK1 associated gene mutations and those patients with PRSS1. PRSS1 patients seem to develop a more progressive lipomatous atrophy of the pancreas, and I know Dr. Shaw alluded to this a little bit in his talk on the pathology or histopathology of the pancreas in CF. And up top, you can see the SPINK1 patients, I think, have the more classic pattern that we all think of, exocrine parenchymal loss with replacement by prominent fibrosis. Almost sounds like alcoholic chronic pancreatitis. So, this can all be a little different based on the underlying gene mutation. My final point is one about trypsin. We know that high levels of trypsin are really synonymous with inflammation. Low to very lows of trypsin are actually quite synonymous with fibrosis. And why is that important? Because fibrosis is really the hallmark for the development of exocrine insufficiency and diabetes. And this is coming from the chronic pancreatitis literature, but it actually nicely then segues into the cystic fibrosis literature. And this was a nice study published in the Journal of Pediatrics now almost 16 years ago, looking at the rate of trypsin levels declining and suggesting that they tend to be more severe, they tend to be more rapid in patients with severe cystic fibrosis compared to those who are pancreas sufficient. And that rapid decline actually inversely correlates with fecal fat excretion. So in other words, when your trypsin levels rapidly decline, it suggests that you've more rapidly developed fibrosis. And once you have that fibrosis, then you're more likely to excrete more fat in the stool. And so you can see here on the two panels, panel A and panel B, you have patients with severe CF, and you've got patients with CF and meconium ileus. And you can see that their rate of decline is much more rapid than when you compare it to the pancreas sufficient CF patients on the far right in panel C. So the key points of my lecture are really what I stated at the outset. Exocrine insufficiency is a syndrome of which one feature is pancreatic digestive enzyme output, but nutritional need, intake, and intestinal adaptation are just as important. CFTR modulators can restore exocrine function in some patients with PICF, but they tend to be younger and they have detectable fecal elastase levels, suggesting acinar cell reserve. CFTR modulators can increase the risk of acute pancreatitis and PICF even without improvements in fecal elastase, suggesting that even small incremental acinar cell reserve is sufficient to cause acute pancreatitis. CFTR modulators prevent recurrent acute pancreatitis in PSCF patients with a history of acute pancreatitis and can reduce hospitalizations in both PI and PSCF. And finally, exocrine function may not recover in all patients due to environmental factors, lack of the CFTR genotype-phenotype correlation, presence of other pancreatitis mutations, and the presence of subclinical inflammation that leads to pancreatic damage/fibrosis over time. Thank you very much for your time and attention.
Thank you very much. I'm going to see if there's... Go for it. You want to go ahead and read my iPad kind of, sorry. Okay. I got it actually. Yeah. Okay. So let's just see. Oh, no, go ahead,
All
sorry.
right. So,
It's changed over
nice
to
talk,
Sorry,
Vigesh.
sorry.
We got two questions. One from Stacy Allen. She asks, has the restoration of the pancreas happened immediately on pediatric patients, or does it usually turn on after a certain amount of time on highly effective modulator therapy? And do you regularly retest fecal elastase after starting a patient on highly effective modulator therapy at regular intervals, or let the patient's symptoms determine testing?
I mean, it's a good question. Certainly, I'm not involved with children, so it's probably a question a little bit more. But if you go by the literature, it seems like it's all over the place. I think if you can start kids early, you may not have to go that long before you start to see some improvements. We are actually going through, as a center, we're going through our pediatric CF data, and what we're finding is, at least in the historical cohorts, we don't have a lot of baseline fecal elastase measurements. So I think moving forward, I think it would be good to do this so we can figure out what value it has, or whether we should be looking for a better marker of exocrine function. So I think that's probably a simple way to answer that. But in the adult population, we probably have more fecal elastase measurements as a whole. I don't know if we've actually looked at seeing, I don't think, to my knowledge, I don't think we've seen it in an adult patient yet that they've actually restored their exocrine function entirely. I think you may be able to preserve it to some extent in the PSCF patients, but I think PICF is going to continue to be a challenge because of the fact that these patients probably have a lot of underlying fibrosis at this point. There may not really be anything to rescue.
Yeah, I agree. I guess as a pediatric gastroenterologist, I can comment that I think that, yes, we do encourage rechecking, although I would agree we also historically have had a paucity of fecal elastases, as often people would have one, maybe two, and never have them rechecked again. So I'm very much encouraging that, although we don't yet have a predetermined timeline to do that. So, and then I think one last question here. Are there any imaging or pathologic changes in the pancreas in those that recover some pancreatic function?
That's a good question. And in fact, I think there is some interest in looking at radiomics to not only diagnose exocrine insufficiency, but if you can come up with radiomic signatures that are correlative, maybe you can actually see if something changes with treatment over time. But I don't think we have anything that is sensitive enough to pick up the changes that might occur in somebody with CFTR modulator therapy, at least at the ultrastructural level of pancreatic imaging.
Right. Thank you, Vigesh. And I'd like to thank all of our speakers. We've had a wonderful session. Thank you for everyone's participation, and it's nice to see so many people here.